Hello and welcome to this new presentation on genetic sciences. First chapter, Introduction to Genetics. Definition. Genetics is the branch of biology that studies heredity and variation in living organisms, which is inscribed in the DNA. It focuses on how genetic information is stored, transferred or transmitted, and expressed from one generation to the next. This information is encoded in desoxyribonucleic acid, or DNA, which contains the instructions necessary for the development and functioning of all living organisms. At the molecular level, genetic information is organized into discrete units called genes. These genes are responsible for determining specific traits and are passed from parents to offspring. History of genetics. The study of genetics has evolved significantly over time. Early theories of inheritance were largely speculative until experimental evidence provided a scientific foundation. Key historical milestones include pre-Mendelian theories, blending inheritance and pangenesis, 19th century establishment of particulate inheritance through Mendel's work, early 20th century rediscovery of Mendel's law and development of chromosomal theory, mid-20th century discovery of DNA, which was a very important breakthrough by Watson and Crick, and late 21st century advances in molecular biology, genomics, and biotechnology. Now, this is the secular point of view on genetics and the development, the historical development of genetics from a secular point of view in the secular sciences. However, if you go back to the Bible, it talks about, even in the book of Genesis, where it speaks about God creating the seed, the plants, the animals, and each reproducing after its own kind, which means that each species reproduces after its own root, after its own kind, or not a gender as in male or female, but after its own category. And not only category, but species type, meaning that dogs can replicate as dogs, cats as cats, okay? There's nothing in the middle, so it goes against evolutionary science, okay? Now, of course, through hybridization, you can alter that, you can tamper with that, but it would be the works of men, not of God, okay? And by the way, since it requires intelligence to alter and to cross two different species, to produce a third kind, which is a hybrid, a mixture of two kinds that don't usually mix. For example, a dog that is half of a cat, yeah, it's doable. But since it requires an intelligence to do that, why did it not require an intelligence to create the original? Which means that, I'm alluding to the existence and the reality of God. God has always existed. Like, God is real, God is true. Unlike many popular point of views today. Okay? So, because it requires intelligence. And also, another evidence is the genetics. Like, the genetics is a code. The DNA is a code, very intricate, complicated, complex code. It could have not been birthed through evolution. This is what I'm trying to say. Okay? Contributions of Gregor Mendel. Gregor Mendel is considered the father of genetics. Through his experiments on pea plants, Rhizome sativum, he established the fundamental laws of inheritance. Mendel introduced the concept of discrete hereditary units, now known as genes, and formulated two key principles. Okay, first key is the law of segregation, which means that each individual possesses two alleles for a given trait. Okay, so for any given physical, biological trait, such as the eye colors or the hair color, or you name it, depending on the species, after all, you have common traits for different species. So you have the color of the eyes for more than only us humans. Okay, so for each feature or each physical trait, you have two alleles. And these alleles separate during gamete formation. So you have A, lowercase a, and they split to capital A or lowercase a. Okay, so the coming generation, the children would have either this capital case A or lowercase a, which would be two different traits. One would be, by the way, either dominant or recessive, and we're going to talk about that. Law of independent assortment, that would be the second law. Genes for different traits assort independently of one another during gamete formation, which is valid for genes located on different chromosomes. Okay? So PAB, meaning the probability of having A and B at the same time, is equal to probability of A multiplied by the probability of B. So the probability of having two traits at the same time is lower than having one trait because it is the product of two probabilities. And as we know that the probability is a value less than 1. So when you multiply it, it becomes lower. Okay? It's less probable for someone to have three traits in a descendants, okay, in the next generation than having one trait because you have to hit the three of them. Now, in this graph here, in this representation, this is how the crossing happens. Okay, so let's say capital A is actually the eyes that are brown, okay? Brown colored eyes. Lowercase a are the blue eyes. Now, capital means that this trait is dominant, meaning that the mom has both the brown eye and the blue eye. However, A, which is the brown eye, is dominant, but lowercase a, which is the blue eye, is recessive. In other words, only the dominant one, which is the brown one, would show up, would appear in the next generation, or actually in the mom. The mother is displaying the brown eye, meaning that her eye is brown, despite the fact that within her genes, she also has the blue eye trait. But since it's recessive, it does not show up as a physical trait, despite the fact that it is embedded within the gene code. Same thing for that. So what would be the probabilities here? You would have A and lowercase a, capital case A, lowercase a, and you multiply them. So you have AA, capital A, lowercase a, capital A, lowercase a, and lowercase a, lowercase a. Only when you have two recessive genes that are blue, you would get a blue eye or blue eyes within the children. Only when both of them are recessive. In any other case, okay, so capital A or capital A, lowercase a, since the capital A, which is the brown eye, is dominant, you would only see brown eyes within the children. Okay, so you have 3 to 1. This is the ratio. You have 3 possibilities of having brown eyes to only 1 possibility of having blue eyes. So in other words, you have 25% probability of having blue eyes only of all of 100%. Branches of genetics. Genetics is a broad field divided into several specialized branches. You have classical genetics, which focuses on inheritance patterns and the transmission of traits across generations using observable characteristics. It relies heavily on Mendelian principles. Second, molecular genetics, which studies the structure and function of genes at the molecular level, so it is more in depth, more accurate, particularly DNA and RNA, ribonucleic acid, and protein synthesis. Okay? Central dogma of molecular biology, you have DNA, RNA to protein or protein. Population genetics examines the distribution and changes of allele frequencies in populations over time, influenced by evolutionary forces. I don't buy into evolution personally, okay? But okay. So as selection, mutation, migration, and genetic drift, allele frequency is given by P plus Q is equal to 1. Hardy-Weinberg equilibrium is given by P squared plus 2PQ plus Q squared is equal to 1, where P is equal to the frequency of dominant allele, Q is equal to the frequency of recessive allele. And you notice here, like the relationship is non-linear between dominant and recessive, okay? Basic terminology. Gene. A gene is a segment of DNA that encodes a functional product, typically a protein. So it is a block of information characterizing a trait or a protein or something within the body. Allele. An allele is a variant form of a gene. For example, a gene controlling flower color may have alleles for purple or white. Okay, so let's say you have two alleles, one purple or one red, one white. Okay, so you have heterogeneous allele where the chromosome would have two strands. One strand would be carrying the red trait, the other is carrying the white trait. Within the same chromosome, okay, two strands, each carrying a different trait, that would be a heterogeneous allele. As for the homogeneous allele, both of the strands of the chromosome would be carrying either white trait or red trait. Both of them, okay? They would be homogeneous. Now, of course, one would be dominant, one would be recessive, or both of them, they could be dominant, okay? Which means that the physical trait resulting from codominance becomes pink, which is the mixture between red and white. Genotype. The genotype is the genetic constitution of an organism represented by allele combinations such as A, capital A, capital A, capital A, lowercase a, or double A, lowercase, okay? Which we've seen in the crossings in a previous slide, okay? When we examine different probabilities for the descendants of a crossing over between male and female in a first generation. Phenotype. The phenotype is the observable physical or biochemical characteristic of an organism, meaning that these are the traits that actually show up. Okay? They are not recessive. They are not hidden. They actually showing up. Determined by its genotype and environmental influences. Okay, so phenotype is equal to genotype plus environment. For not all flesh is the same, but there is one kind for humans, another for animals, another for birds, and another for fish. That's 1 Corinthians 15:39 in the Bible. Denoting that for each kind, okay, you have a different kind of DNA, a different kind of, and also a different pattern, not pattern, okay, but the thing is, in reproduction, it would reproduce in the same way. It doesn't reproduce something else. So, in other words, the claim that fishes came from birds or from whatever, this is not actually true. Each kind reproduces after its own type, after its own species. It cannot produce something different, a new species. There is no crossing over like that. Unless you force it in a lab, that would be a completely different discussion. Okay? But God created everything to reproduce after its own kind separately. The fish were created, the birds were created. Now, within the same species, you could have what you would call evolution, meaning that within the same, like you have a generic type for birds, okay, from which you can get different types of birds, different species of birds, okay, but they come from the same prototype. This is what explains how on the Ark of Noah all the animals could be gathered, okay, because you had the prototype gathering on the same ark. But from the same prototype, okay, like from the tiger, you could have different types of cats. They come from the same parent. And all of these DNA traits were already stored within the genes. This is, by the way, what explains that. How you can have as many types of hair color on the earth. The genetic information was already existing in the first man, which is Adam. From that, you have, dominance, recessivity, you have mixture between blonde and black hair, etc. This is how you get infinity of colors when it comes to the hair. All right? This is how you get the infinite colors in the eyes. So this is what explains it, meaning that the genetic information was already built in, and then through reproduction, through different descendants in the families. You have some traits showing up in one generation, other traits being recessive, they show up in the next, etc. Overview of heredity. Heredity is the process by which traits are transmitted from parents to offspring through genetic material. This transmission occurs via gamete, which is sperm and egg, each carrying half of the genetic information of the parent. Okay? So during fertilization, you have one gamete from the first parent, from the male, another from the female. Both of them, they are crossed together, like they attach together to form the zygote, 2n, which is within the baby, where n is the haploid number of chromosomes, 2n is the diploid number of chromosomes. So two strands of chromosomes, each coming from one parent, they stick together to form, the double-stranded chromosome in the baby, in the next generation. Heredity ensures both the continuity of species and the introduction of variation, which is essential for evolution, so to speak, and adaptation. It's interesting how the science of genetics appears in the Bible in multiple places. One of them is the book of Numbers, verse 14 to 18, where it says that the Lord is slow to anger and filled with unfailing love, forgiving every kind of sin and rebellion. But he does not excuse the guilty. He lays the sins of the parents upon their children. The entire family is affected, even the children in the third and fourth generations. That's Numbers 14:18. Now you might ask, how? You see, sins are not something only spiritual. It spills over to the body, to the physical traits, okay? So it can be a curse of sickness, something like that. Like there's a harvest with sin, meaning that just like in the Bible it says the wage of sin is death, there are consequences even in the body, and they are transmitted in the genetics from one generation to another. Here it says it can be all the way, like also on another translation it says that the Lord visits the sins of the parents in the children to the fourth generation. Now, how is that? This is how it works out in the science of genetics. So let's consider that the curse or the sickness or whatever is being transmitted, which is the consequence of sin within the genes of a family, is designated by the blue color. Okay? So you have a parent who carries a curse, a sickness, whatever it is, and he marries someone who is supposedly clean from that very thing. Okay? So this is how it breaks down. This would be, of course, the first generation. The second generation, you would have 50% of this trait being transmitted to the second generation. Okay? Now, because each descendant or like each child would get 50% of the traits of the family, okay, of the parents. Now, this second generation, okay, all of the children have this kind of curse, whether in the light blue or the dark blue. You take someone from the second generation, he marries the second time, okay, not the second time, he marries to produce the third generation. So you get here, this 50% becomes 25%. Why? Because this 50% that he has here from the generation before him, it gets broken down into, he gives from it half, which is 25% to the next generation. So we have 25% here, 25% here, and two kids were not affected by this curse or this transmitted disease. All right? So that would be the third generation. All the way to the fourth generation. Why is that? Because this guy here can also marry again, correct? So he would get married again and produce also still children that carries the same sickness. But by the fourth generation, it would have dissolved. It would have been dissolved. All right? Meaning that by the fourth generation, you would have no trace of it. So this is what it means that he visits the sickness or like the sin of the children all the way to the fourth generation. Now let's talk about the cell structure and chromosomes. Okay, So you have the ribosomes. These are the ribosomes. The function, it is the cell's protein factories. So they produce a protein, translating RNA into polypeptides. So the RNA goes and it copies the info from the DNA in order to fabricate or manufacture protein, okay? Or rather, like they start with polypeptide in order to synthesize protein. Golgi apparatus, its main purpose is processing, sorting, and packaging. So the function is, okay, this is Golgi's apparatus. So it's like a post office. It receives protein, okay, and lipids from the RER. It modifies them according, to carbohydrate groups to make glycoproteins. It stores them, packages them, then, into vesicles. So the main purpose is processing, sorting, and packaging. Lysosomes, this one here, intracellular digestion and recycling. Function, lysosomes act as the cell's digestive system or waste disposal center. They are membrane-bound vesicles containing acidic hydrolytic enzymes, okay, lysosomes that break down damaged organelle macromolecules and foreign invaders like the phagocytosis. As for the mitochondrion or mitochondria is like the power plant to the cell. It is like the energy manufacturing or production center for the entire cell. Okay? So every energy that the cell needs is being generated by this power plant, which is the mitochondria. Now, for the nuclear pore and the nucleus is where the chromosomes are located, and the chromosomes are actually the bundles of the DNA which carries all the information for different proteins within the body, for different physical traits that you would see, and even the ones that are hidden, that you do not see. So every type of information needed to manufacture something or replace a cell or reconstruct a damaged organ are located within the chromosomes and specifically within the bundles of the DNA molecule. Chapter or Module 2, Cell Structure and Chromosomes. Overview, genetic information in living organisms is stored, organized, and transmitted at the cellular level. Understanding cell structure and chromosome organization is fundamental to explaining how DNA functions, replicates, and is inherited. Another verse from the Bible in Genesis 1:24, and God said, "Let the earth bring forth living creatures according to their kinds, livestock and creeping things, and beasts of the earth according to their own kinds," meaning their own species. Okay, another evidence, and by the way, if you see, for example, a bicycle and a car next to each other, this doesn't mean that the bicycle came from the car or the car from the bicycle. It means that the same designer is behind both. Okay? I heard that analogy once on YouTube, but it makes sense. Okay? It doesn't mean if they have similar traits that one came from the other, because both they are complex and intricate. Now, it can mean also, okay? Not it can mean also, but when you see different types of bicycles, yes, they are coming from one prototype, okay? As for the organism, you had like the genetic modification, the genetic, like in the descendants, you have different crossings between different genes, okay, that can produce different hair colors, different eye color, in the same way that you have different variations of birds coming from the same parent. This you can say, yeah, this you can call as evolution, but it is within the same framework of the same kind, not from something else. Okay, so a bird cannot have been evolved from a fish. They are two completely different designs. Okay, and there's no intelligence in the being itself. It is the designer behind him, not the being, not the creature itself. There is no such thing as natural selection, because actually if you expose a plant or whatever, like a being, an animal, to radiation, you would cause it to grow cancerous cells. Okay, you would kill it. And this random mutations actually destroy genetic information, not build it up. So, okay, you would argue, but perhaps after multiple generations, things will change. You see, when it comes to flies, the fly can have hundreds of generations in a very short amount of time. So you can test on that. And after they had tested radiation, okay, radioactive matter over these the flies, okay, because since they reproduce very fast, what happened is that instead of growing into something else, instead of developing new traits for like a new type of species, what happened is the destruction of the DNA information that was in them. So they started growing up like wings within their eyes. They started growing up wings, or eyes within different parts of their bodies, and they could no longer fly, they could no longer function normally, okay? And this resulted gradually in the death of the next generations. So it cannot work like that. That's the point. Cell types, prokaryotic cells. Prokaryotic cells are simple unicellular organisms that lack a membrane-bound nucleus and organelles. Their genetic material is typically found in a single circular DNA molecule located in a region called the nucleotide, okay, or nucleoid. Key characteristics: no true nucleus, circular DNA, absence of membrane-bound organelles, and very small size, approximately 1 to 10 micrometer, which is 10 to the power of minus 6 meters. The genome size, example relation, so the length of the DNA is directly proportional to the number of genes. Okay, so they are both related. Here is an example of a prokaryotic cell, like the bacterial cell anatomy. You have cytoplasm, which is the body within the cell, pilus, capsule, cell wall, which is the outer skin, plasmid, and nucleoid DNA, okay, which is like a circular DNA structure. And pigtail, by the way, or flagell, which enables the bacteria to travel, like to swim. And by the way, the mechanics behind that are very complex. They could not have been birthed by evolution because they looked on a micro level on the shape of these gears. It's like a gear, okay? It is the same gear that you have in your car at the micro level present in a bacteria, which was supposed to be the very simple cell, okay? The most basic cell structure or architecture. But meanwhile, if you look deep into it, you have advanced mechanical principles that allow the bacteria to travel, to swim. It's not as simple as people think. No, there is a very genius creation behind that. Eukaryotic cells, just like the cell example that we've seen before, eukaryotic cells are more complex and contain a membrane-bound nucleus where genetic material is stored. DNA is organized into multiple linear chromosomes. This is what we referred to before, and this is the same structure that we've seen at the beginning of this chapter. Key characteristics: presence of nucleus, which is like the inner core of the cell, linear DNA molecules, again, they are bundled in the form of chromosomes, membrane-bound organelles, and larger size going up to 10 or from 10 to 100 micrometers. Again, this is the Golgi apparatus within the cell, which stores protein. Okay, it's like a storage warehouse. The mitochondria, which is the power plant for the cell, it generates the energy that the cell needs. The lysosome is like the recycling and the disposal of waste within the cell, and the ribosome, which are necessary to produce protein. So they are the manufacturing plant for the protein production, okay, through the RNA, which copies the DNA in order to synthesize the protein from the polypeptide. Structure of the nucleus. The nucleus is the control center of the cell and houses the genetic material. Main components, you have the nuclear envelope, which is a double membrane separating the nucleus from the cytoplasm, which is the inner core of the cell, from the cytoplasm, which is most of the volume of the cell, okay, where you have the mitochondria, the lysosome, ribosomes, and the Golgi apparatus. Anything that serves the function of the cell outside of the functionality of the genetic information handling, okay? Now, the nuclear pores, which are the interface between the nucleus and the cytoplasm, they are necessary for molecular transport. Nucleolus, which is the site of ribosomal RNA, rRNA synthesis, okay, to synthesize or produce proteins, and chromatin, which is the DNA protein complex. DNA content in a diploid nucleus. DNA content is given by 2n multiplied by L length of haploid. Cell duplication or mitosis. You have another type, which is meiosis. We're going to get to that also. Now, mitosis is the replication of a cell from its own material. Okay, so it splits into two replicas, two copies. This is how it works. Prior to mitosis, each chromosome makes an exact duplicate of itself. Okay, so each chromosome splits into two chromosomes as a duplicate. Then chromosomes thicken and coil. In early prophase, the centrioles, which have divided, form asters, okay, and move apart. The nuclear membrane begins to disintegrate, which is the nuclear level cover, okay? Now, in late prophase, the centrioles, which are these two here, are at opposite poles. The nucleus or nucleolus and nuclear membrane have almost completely disappeared. They start like a weaken as a preemptive step before splitting, before breaking apart to form two cells. The doubled chromosomes, their centromere attached to the spindle fibers, line up at mid-cell in metaphase, like this, here. Okay, in early anaphase, the centromere split, half the chromosomes move to one pole. This is here, yeah, half moves to one pole, another move to the other pole. In late anaphase, the chromosomes have almost reached their respective poles. Here also, the cell membrane begins to pinch at the center. This is when it started to weaken. The cell membrane completes constriction in telophase. Nuclear membranes form around the separated chromosomes, which is here. At mitosis completion, there are two cells with the same structures and number of chromosomes. Same exact copies as the parent cell. As for the meiosis, it is when you have a crossing between a male and a female to produce a new generation. This is where the meiosis come into play. So you have at first two strands of the chromosomes. Of course, from each chromosome you have one strand splitting, okay, in order to be crossed with the other strand. So here you have a cell with two pairs of chromosomes, okay. Now each pair crosses with the other pair, with one strand of the other chromosome. Crossing over leads to DNA exchange, and here you start getting different versions, like different chromosome flavors, so to speak. You have here, this one is formed of the crossing between these two, four different gametes. Okay, basically four different gametes. This one, this, and this. Okay, cell divides a second time here, forming this one and this one. Chromatin versus chromosomes. Chromatin is the less condensed form of DNA found during interphase, allowing gene expression and replication. Chromosomes are highly condensed structures visible during cell division. So basically, the chromosomes are the packing or the packaging version of the DNA. The packaging status okay, or condition of the DNA. Condensation relationship, chromosome is equal to condensed chromatin. Levels of compaction, you have DNA double helix, 2 nanometers, nucleosome fiber, 10 nanometers, chromatin fiber, 30 nanometers, and loop domains. Okay, and you have fully condensed chromosomes. Karyotype. A karyotype is the complete set of chromosomes in an organism, arranged by size, shape, and number. Human karyotype is given by 2n, which is equal to 46, equal 44 autosomes plus 2 sex chromosomes. Abnormalities can be expressed as 2n plus minus k, where k represents the number of missing or extra chromosomes within the organisms, for example, the trisomy. DNA packaging. DNA must be efficiently packed to fit inside the nucleus while remaining accessible for transcription and replication. Transcription is when the RNA copies parts of the DNA in order to manufacture protein, okay, and replication. Replication, so also in order to replicate the cell. Histones are positively charged proteins that interact with negatively charged DNA. Electrostatic interaction is given by F is proportional, the force of interaction, proportional to qDNA times the quantity of histone divided by R square. In other words, the force of pull to the charge is proportional to charge of the DNA multiplied by the charge of the histone divided by R square, which is the distance between the DNA and the histone square. Okay, so it is the distance between the DNA molecule and the histone, which are the positively charged proteins, square. Nucleosomes. The nucleosome is the fundamental unit of DNA packaging structure. DNA wraps around a histone octamer, approximately 147 base pairs per nucleosome. Wrapping can be approximated as L wrapped, the length of wrapping, approximately 1.7 multiplied by 2 pi r, where r is the radius of the histone core. Compaction ratio, how much compact this structure becomes, is equal to L extended DNA divided by the L packaged DNA. In other words, how much the compaction has reduced the length of the extended DNA, okay, after compaction. How much it has been compressed in order to pack it around the histone. Genome organization. The genome refers to the complete set of genetic material in an organism. Types of DNA sequences. You have coding DNA, which encodes the proteins. Non-coding DNA, which is for regulatory purpose. Introns, repetitive sequences. Regulatory sequences, which are promoters and enhancers. The gene density is given by number of genes divided by the length of the genome. This is how you get the gene density, meaning that how many genes are there divided by the length, okay, for a specific trait. Euchromatin versus heterochromatin. Euchromatin, less condensed, transcriptionally active. For transcription, this is when the RNA goes within the double helixes of the DNA, which, by the way, opens up. It kinds of partially splits, and the RNA goes within in order to transcribe, in order to copy the DNA for protein manufacturing. The heterochromatin, highly condensed, transcriptionally inactive, unlike the euchromatin. Expression relationship, the gene expression is proportional to 1 divided by the chromatin condensation. The genome size variation, genome size varies widely among organisms and does not always correlate with organism complexity. So it's not always a function of how much complex the organism is. Just like, by the way, the theory of information. Sometimes small blocks of information can describe a very complex structure. Okay, just like data compression when we talk about the digital application. Okay, so this is C-value paradox called. So the genome size is different than the organism complexity. It's not proportional to it. Finally, the structure of the cell and organization of chromosomes are essential for understanding how genetic information is stored, regulated, and transmitted. From DNA packaging into nucleosomes to large-scale chromosome organization, these mechanisms ensure both efficient storage and functional accessibility of genetic material. Chapter 3, DNA structure and functions. Deoxyribonucleic acid, or DNA, is the fundamental molecule that stores genetic information. It is like a USB flash drive that stores data. In all living organisms, its structure enables accurate replication and transmission of genetic information from one generation to the other. Also, replication in order to conserve the cells, okay, to keep backups for the genetic info, while its sequence encodes the instructions necessary for cellular functions and development. As you notice here in this graph, in this display to the right, okay, all the way. So chromosomes basically are the bundling of the DNA molecule. Okay, the DNA molecule, if stranded, if it's split, you have two helixes that around each other form a spiral-like architecture where you have a pairing of different acids, and these acids pair in a ordered way and a unique way, meaning that you have 4 bases that are adenine, guanine, cytosine, and thymine, or ACGT. A pairs with T, adenine with thymine, and cytosine with guanine. Okay, how they pair and their sequences is what codes DNA information. Just like in informatics, you have zeros and ones, which are 2 bits, 0 or 1. These are what is used in order to code information in computer systems. Here you have like four possible states: adenine, guanine, cytosine, and thymine, and how they are pairing. How is the sequence made up? This is what codifies the information of the organism. Discovery of DNA structure. The double helix structure of DNA was proposed in 1953 by James Watson and Francis Crick, based on experimental data from X-ray diffraction studies conducted by Rosalind Franklin and Maurice Wilkins. This discovery revealed DNA consists of 2 complementary strands paired with each other. The strands are arranged in a helical structure like a spiral, and base pairing is specific and essential for replication. Double helix structure. So the DNA is composed of 2 anti-parallel strands forming a right-handed double helix. Key geometric properties: helix diameter is approximately 2 nanometers. Distance between base pairs approximately 0.34 nanometers. One turn approximately is equal to 10 base pairs. Helical pitch, P is equal to 10 multiplied by 0.34 is equal to 3.4 nanometers. Each strand has directionality, which means that 5' goes to 3' and 3' to 5'. Nucleotides or nucleotide. DNA is a polymer made of repeating units called nucleotide. Each nucleotide consists of a phosphate group, a deoxyribose sugar, and a nitrogenous base. The 4 nitrogenous bases in DNA are adenine, A, thymine, symbolized by T, cytosine, and guanine, G. General nucleotide representation: the nucleotide is equal to phosphate plus sugar plus base. DNA strand formation via phosphodiester bonds. You have the sugar bonding with phosphate with sugar I plus 1, okay, on both sides. Base pairing rules: the 2 DNA strands are held together by hydrogen bonds between complementary bases. Base pairing rules: A pairs with T, or adenine with thymine, and cytosine with guanine. hydrogen bonding, so AT you have between A and T, 2 hydrogen bonds, between C and G or cytosine and guanine, 3 hydrogen bonds. Chargaff's rule, you have A bonding with C, adenosine with thymine, and C with G, or cytosine with guanine. Total base composition, A plus T plus C plus G is equal to 1, which means, in other words, that the probability of having one of these, okay, either of these 4 bases in a DNA chunk, okay, is equal to 1. Because definitely you're gonna see one of these because these 4 constitute the entire DNA structure. GC content, which means the content in guanine and cytosine in a genotype, for instance, in a DNA block or code, is equal to sum of guanine acids plus cytosine acids divided by the sum of all types of acids within the DNA block, within this information block, which is A plus T plus C plus G, multiplied by 100. So, in other words, let's say we have this sequence here, G, C, G, A, T, G, C, A. Okay, so the percentage in guanine and cytosine is equal to how many G or C are there? You have 1, 2, 3, 4, 5, divided by the total, which is 5 plus 6, 7, 8. 5 divided by 8, okay? Multiplied by 100, this gives you the percentage of GC. All right, so this is more than 60%. DNA versus RNA. DNA and RNA are nucleic acids with distinct structural and functional differences. So for the DNA, it is double-stranded. So this is the double helix acid that you see usually in pictures, contains deoxyribose sugar, uses thymine, T, and it is used for long-term storage of genetic information within cells, within specifically chromosomes. The RNA is similar to the DNA, but it has a different structure and functionality. For the RNA, it is single-stranded, so it is one strand of the DNA, contains ribose sugar, so structurally it's also different, uses uracil instead of the thymine, okay, so it is different. And this is usually during the transcriptase, okay, the transcription of the DNA, you have the RNA goes in the midst of the DNA strand, which splits into partially strips, allowing the RNA molecule to enter in its midst in order to copy the DNA. However, during the copying, okay, so the adenine, it copies it into okay, uracil. It uses, in fact, uracil instead of the thymine. This transcription substitution rule, so during the transcription, the adenine is copied, actually not copied, it is transcribed, meaning that when the DNA has the RNA going into the midst of the strand, it copies A into U. So it doesn't exactly copy, but it creates the match. However, the match within the RNA is not the thymine, it is the uracil. Now, for the thymine, it is transcribed into adenine. For the C and G, meaning cytosine, guanine, guanine, cytosine. So everything is the same except for the adenine is transcribed into uracil. Central dogma of molecular biology. The central dogma describes the flow of genetic information within a biological system. So how does the genetic information flow? You have the DNA, which is the storage place for the genetic information. Again, as we mentioned before, the DNA is the molecule that stores genetic information within the cell. Then the RNA goes and transcribes the DNA. Okay, and after this transcription, you have a conversion into polypeptide. So this is used in order to fabricate polypeptide, from which you have the synthesis of protein. Transcription, the DNA is transcribed into messenger RNA or mRNA. Okay, this is what the transcriber is called. Messenger RNA molecule. If DNA sequence is DNA 3' TACG, okay, you have the messenger RNA sequence becomes, it is like the flipping of it, meaning that the thymine, as we've seen before, is transcribed into adenine, adenine into uracil. This is the only distinction or exception here. Other molecules or other acids remain the same, meaning that the C is transcribed into G, which is the flip side of it. G is transcribed into C. Okay, so thymine, adenine, cytosine, guanine, guanine, cytosine, except for the adenine is transcribed into uracil. This is the main difference. After that, translation. mRNA or messenger RNA, in French, by the way, they call it ARN transcriptase, okay, the transcriber of RNA. mRNA is translated into a protein sequence using codons, which are triplets of nucleotides. Number of possible codons is 4 to the power of 3, which is 64. 4 being the 4 bases, correct? To the power of 3, so it is 64. Protein length relation is given by number of amino acids is equal to the number of nucleotides divided by 3. And 3 is due to the fact that they are triplets. That's why. Here within this graph or illustration, we see that the DNA splits in half, and this is where the RNA, mRNA, goes in the midst in order to transcribe. Okay? So it starts not copying, transcribing, meaning that it gets the opposite of each amino acid. So again, A is transcribed into uracil, thymine into adenine, etc. You have the RNA primer, DNA primase, DNA ligase, DNA polymerase, etc. Okay, this is the direction of transcription. You have the lagging strand and the leading strand. Okay, lagging strand meaning here the RNA goes below the DNA molecule. Here it is the leading strand, meaning that, yeah, you have, this is the DNA here, the RNA is leading, so it is on top. Okay. Functional role of DNA. DNA encodes genetic information that determines protein synthesis, cellular structure and function, hereditary traits. Information content can be expressed as I, number or total number of information is given by n log base 2 of 4, okay, is equal to 2 multiplied by n bits, where n is the number of nucleotides. Here another illustration for the DNA structure. So you have the pairing between adenine, thymine, guanine, cytosine, and that would be the DNA bundled into a chromosome, which is for the storage on the inside of the cell, storing DNA and inheritance information within the cell. Of course, as we notice that it is made of a double helix, and by the way, this is so significant because the helix so much look as a sound wave, which means that this DNA structure within the human body is designed so as to respond to sound frequencies. And there are many proofs to that. In the Bible, there's a verse that says, as a man thinks in his heart, so is he. Another verse, that you shall eat the fruit of your own lips. Okay? Also in the book of James, it says that the mouth is like a rudder, meaning that what you think, you will go. So it will take you places. So whatever you declare shall be established unto you. If you're declaring something over your life, if you're saying, okay, I'm going to do this, I'm going to do that, I'm going to succeed, I declare and decree in Jesus' name that I'm going to travel to this place, do that. Okay? So you are steering your life down that direction. But at the same time, it can be so damaging, because if you're proclaiming death on your life, if you're proclaiming that nothing would change, like things won't work out, you'll end up eating the fruit of that. This is why doubt is dangerous, because you start speaking death over your life, over your destiny, and the calling that God has over your life. And it will wear you down, and it will start dragging you backwards and hindering what God has for you. So what we say, it affects our entire being. Plus, there was another experiment on water where they've seen that when you say something over the water, like when you have a repetitive word on a broken record over the water will start changing in structure, and even it can become either polluted if there's a curse word being spoken over the water. They tested it by actually freezing the water, and they looked down in a microscope. They saw this crystal shape like Christmas decoration. They saw it either being damaged or forming into a beautiful diamond shape, depending on whether a good word or a bad word is being spoken. Plus, if the water is putrid, it becomes cleaner or starts becoming contaminated if there's a curse word. Like when they played hard rock over the water, it became like a black hole. The look of the water became so hideous. So what you say, what you speak, either transform you in a good way or in a bad way. Faith comes by hearing, and by hearing of the word of God. Another verse, do not be conformed to the patterns of this world, but be transformed by the renewing of your mind. These all biblical scriptures. So what you think about this is the very thing that you will manifest. That's why it's important to feed on the word of God. This is how you start transforming on the inside, and you've been given all that we need for life and godliness. In Deuteronomy 8:18 it says, it is God who gives you the power to produce wealth. So it is about changing on the inside before everything outside starts changing for you. It is a matter of faith and believing something before this something can actually start manifesting in your life. Finally, the structure of DNA is intricately linked to its function as the carrier of genetic information. From the double helix configuration to precise base pairing, DNA ensures accurate storage, replication, and expression of genetic instructions, forming the molecular basis of life. Chapter 4, DNA Replication. Okay, DNA replication is the biological process by which a cell duplicates its genetic material prior to cell division. This process ensures that each daughter cell receives an identical copy, the same material of the original cell, especially the genome, the genetic information. Replication is highly accurate and tightly regulated. Semi-conservative replication. DNA replication follows a semi-conservative mechanism, meaning that each newly formed DNA molecule consists of one parental template strand and one newly synthesized strand. Okay? If the original DNA molecule is the DNA parent equal to S1, S2, then after replication, DNA1 would have S1, S1 prime, and DNA2 would have S2, S2 prime, meaning that it takes half of the original information of the parent, which is 50%. Where S1, S2 are parental strands, S1 prime, S2 prime are newly synthesized complementary strands. Complementary condition, you have Si prime is equal to the complementary of Si, the complement, the completion of Si. Replication fork and directionality. Replication begins at specific sequences called origins of replication and proceeds bidirectionally in both directions, forming replication forks. DNA synthesis occurs in the 5' to 3' direction. New strand growth is proportional to 5' to 3'. To make it a little bit clearer, this is the direction from 5' to 3'. This is the directionality for the replication. Okay? So the new strand growth is from this point up to this point in this direction. Key enzymes in DNA replication. You have at first the helicase. Helicase unwinds the double helix by breaking hydrogen bonds between complementary bases in order to open up the DNA strand. You have the energy requirement is given by ATP, adenosine 3-phosphate, by the way, would give us adenosine diphosphate plus pi phosphate plus energy. Unwinding rate can be approximated as U unwind, okay, unwinding ratio is equal to delta NBP divided by delta T. In other words, this is the number of complementary bases that have broken up in respect of time. This is the ratio. DNA polymerase. So DNA polymerase synthesizes the new DNA strand by adding nucleotides complementary to the template strand. So here you have the existing strand added to the deoxynucleoside 3-phosphate would give us a new strand plus pyrophosphate. Rate of synthesis is equal to the number of bases being copied in respect of time. DNA ligase joins discontinuous DNA fragments by forming phosphodiester bonds. Okay, so they recouple or rejoin the DNA fragments. Ligation reaction is given by first fragment added to the second fragment of DNA. They both become the joint DNA, which is the double helix, the classical double helix that we know about already. Leading versus lagging strands. Due to the anti-parallel nature of DNA strands, replication occurs differently on the two strands. You have the leading strand. The leading strand is synthesized continuously in the same direction as the replication fork movement. Okay? Continuous synthesis, and this is, of a high frequency. The lagging strand is synthesized, and we've seen that earlier, is synthesized discontinuously in short segments opposite to the direction of the replication fork. So discontinuous synthesis would give us fragment formation. To make it a little clearer, I reinstated this graph, this illustration. So we'll start with the leading strand. For the leading strand, it is synthesized continuously in the same direction, you see here the arrow, as the replication fork or replication fork movement. You have here a continuous synthesis with a high efficiency, and it is a copy that is continuous, not block-based or chunk-based. Meanwhile, for the lagging strand is synthesized discontinuously in short segments, which is opposite to the direction of the replication fork. So this is the direction of the replication fork, correct? You see, for the lagging strand is going in the opposite direction from right to the left, and it is fragment formation based. So its strategy is to form fragments, not continuously, but fragment based. So it is discontinuous synthesis. Okazaki fragments, short DNA fragments synthesized on the lagging strand are called Okazaki fragments. The fragment length is given by approximately 1000 to 2000 BP prokaryotes. Total lagging strand length is given by L total is equal to sigma I equal 1 to n Li, which is the sum of all strands, all blocks or chunks. After synthesis, RNA primers are removed and fragments are joined by DNA ligase, which is the process where the DNA double strands are formed or joined into one helix. Okay, the double helixes are joined together in a spiral to form the DNA complete molecule. Replication accuracy and proofreading. DNA replication is highly accurate due to complementary base pairing and proofreading mechanisms as justification and error correction. Okay, so error rate without proofreading is approximately 10 to the power of -5. But with proofreading, it is approximately 10 to the power minus 9, which is much lower. DNA polymerase proofreading function. So you have 3' to 5'. This is the direction, the exact opposite. Exonuclease activity. Error correction is equal to number corrected divided by total number of errors. Mismatch detection probability, probability of correct, okay, is equal to 1 minus the error. This is the probability of having correct bits of information. It is 1, which is 100%, minus the error probability. Fidelity of replication. The overall fidelity depends on multiple factors. You have base pairing specificity, proofreading by DNA polymerase, post-replication repair mechanisms in order to make sure that anything that has been replicated, if there's any error or any wrong dispositions within the DNA, they need to be rectified. Total mutation rate is mu equal to epsilon times the number of bases, which is error rate multiplied, error percentage multiplied by the number of bases that have been copied or replicated. Conclusion: DNA replication is a precise and efficient process essential for life. Through semi-conservative duplication, coordinated enzyme activity, and proofreading mechanisms, cells ensure accurate transmission of genetic information across generations. Finally, for chapter 4, we have a beautiful illustration that kind of brings everything together, all the concepts that we've explained before, in one illustration. So you have the helicase, which unwinds the DNA double helix, so it breaks it apart. It breaks the double helix apart in order for the transcription to take place. You have primase, okay, which synthesizes the RNA primer, correct? You have the DNA polymerase, which adds the nucleotides here within the strand, and the ligase, which seals the gaps in order to form, like, it seals the gaps between both DNA strands in order for it to, it closes back those DNA strands into one DNA spiral double helix into the original form. So you have the helicase unwinds the DNA, the two strands, it breaks them apart for the transcription to take place. Then after the transcription has taken place in this area, you have the ligase that seals it back together, those two strands together, to regain or to restore the original DNA double helix architecture or form or shape. We notice also in this picture what we call the Okazaki fragments, which are fragments of the DNA being copied. You have the leading strand here, okay, going in the same direction of the fork. Two identical DNA molecules are being formed, all right, and these the different acids or bases. You have adenine, thymine, cytosine, guanine, okay. You have ATCG and the uracil. During the transcription, by the way, the adenine is mapped into uracil. Don't forget that. Chapter 5, Transcription and Translation. Okay, gene expression is the process by which genetic information stored in the DNA is converted into functional products. Primarily protein. So it is the protein being synthesized based on the genetic information. It is like the recipe for the protein being manufactured by the cell. And by the way, it is the ribosome that actually manufactures the protein, as a reminder. And the Golgi apparatus stores the proteins and digests them and break them down. The lysosome is like a garbage disposal cell, or like a not cell. It is a a periphery within the cell that actually digests and cleanses and disposes the cell from garbage or poison or whatever. So now going back, this occurs in two main stages. You have the transcription, meaning that the protein manufacturing occurs in two main stages. First, transcription from DNA to RNA, then translation from RNA to protein, following the central dogma of molecular biology. So you have three stages: DNA being transcribed by the RNA to copy. Actually, it doesn't copy again. As a reminder, it flips adenine into uracil, thymine into adenine, cytosine, guanine, etc. And then it uses that in order to manufacture protein. Transcription, the general mechanism. Transcription is the synthesis of messenger RNA, mRNA, from a DNA template. It is catalyzed by the enzyme RNA polymerase. DNA template strand you have from 3' to 5'. mRNA synthesis direction is from 5' to 3'. So two different directions on a molecular level. This is what we mean by that. As a reminder, we take another look at what 3' to 5' means. So this direction would be during or for the DNA template strand from this point up to this point on a molecular level. As for the messenger RNA synthesis direction, the synthesis is from here to here, from the 5' end to the 3' end on a molecular basis. So they go opposite to each other. This is what you need to know here. RNA polymerase. RNA polymerase binds to specific DNA sequences and catalyzes RNA synthesis. Polymerization reaction is given by, you have the first DNA strand. It reacts with RNTP, which is the ribonucleoside triphosphate. It gives us another new strand of DNA, new DNA strand, plus pyrophosphate or PPI. The transcription rate, or the rate at which the DNA is being transcribed, in other words, the number of transcribed bases in respect of time. This is what the rate means. It's given by U transcription or new transcription, that's a Greek letter new, is equal to dNRNA divided by dt, or the number of bases that are being transcribed in respect of time. Promoters and initiation. Promoters are specific DNA sequences where RNA polymerase binds to initiate transcription. Binding affinity is given by p-bind proportional to 1 over kd. That's the probability for binding, where kd is the dissociation constant. Dissociation is when the DNA double strands break or separate for the RNA molecule to go in and to transcribe the DNA strand. Elongation. RNA polymerase moves along the DNA template. Add complementary RNA nucleotide. Base pairing during transcription is given by, as we've seen before, the adenine transcribes into uracil, that's the only exception, thymine into adenine, cytosine into guanine, guanine into cytosine. Termination. Transcription ends when RNA polymerase encounters terminator sequences. Termination efficiency is given by eta is equal to number of terminated transcripts divided by number of initiated transcripts. This is another representation. You have the unzipping or unwinding, just like a zipper, which is done by the helicase substance, correct? Helicase molecule. So it breaks the DNA double helix apart for the RNA to be formed, to be copied. The RNA polymerase is the one that creates the RNA and transcribes it, okay? And the RNA would transcribe this DNA to form a, you have the formation of a messenger RNA, which is, by the way, becomes ready for translation, meaning that it needs to be flipped back into the original mapping so that it can be stored as a DNA molecule within the chromosomes. mRNA processing in eukaryotes. In eukaryotic cells, the primary transcript undergoes modifications. First, you have the 5' capping, which is the capping of the beginning of the molecule. Polydenylation, which is the tail processing of the molecule. We're going to explain this further in the next slides. And splicing, which is the removal of introns. To splice means to join. Now, the mature mRNA length resulting from that is equal to L mature is equal to L primary minus the length of introns that needs to be removed, before the splicing process. Starting from the 5' capping. Shortly after transcription begins, the 5' end of the pre-mRNA is modified in order to protect it. Okay, so the modification, a modified nucleotide called the 7-methyl guanosine, M7G, is added to the 5' end, to the 5' head rather. Okay, the functions is to protect the mRNA from enzymatic degradation during the transcription process. Okay, to protect it from being digested or decayed, facilitates export of messenger RNA from the nucleus. Okay, so it's like an identifier head, and enables recognition by ribosomes during the translation so that it can be recognized or identified by the ribosomes so that the translation process can begin in order for the mRNA to be translated into the protein. Polydenylation at the 3' end, which is the tail of the mRNA, a long chain of adenine nucleotides is added. The modification, a poly-A tail consisting of approximately 50 to 250 adenine residues is added. The functions is to enhance mRNA stability, protects against also degradation, it's like a lower capping, and assists in nuclear export. It's like when you have data transmission, by the way, when we talk about packet networks, you have a header and a tail for each packet, like in IP, in networking. When you transmit data over a network, you have a capping by a header and a footer that delimits or that mark the beginning and the end of an IP packet, which carries data. The same process is going on here, not only for identification, but also for protection from degradation of the mRNA molecule. Splicing. Pre-mRNA contains two types of sequences. We have the exons, which are the coding sequences to be retained, and the introns, or non-coding sequences that have to be removed. So, in order to represent it in a graph, these are the exons presented by these rectangles, and the introns would be like this, like skeleton. Okay, these introns have to be removed before the splicing, because once they are removed, we can splice these three rectangles, which are the blocks, the information blocks, together. Okay, so exons are the coding sequences that need to be retained and spliced together, while the introns have to be removed. These are the introns. They are like fork-shaped placement. Now, the process, introns are removed and exons are joined together by a molecular complex known as spliceosome. The result, a continuous coding sequence that can be translated into a functional protein. Final structure of mature mRNA. After processing, the mature mRNA has the following structure. You have the header, which is the 5' cap, the poly-A tail, which is the footer, so to speak, having the signature, and in between you have the exons, which have been spliced to form the full code sequence of the mRNA. After, of course, the removal of introns, as we've seen in the previous example. The conceptual analogy is step 1, you have biological process for 5' capping. The analogy for that is adding a header or a title. 2, poly-A tail, adding a signature or a footer by which this mRNA can be recognized by the ribosome in order to fabricate or manufacture the protein, and you have the splicing of the exons in between after the removal of the introns, editing out unnecessary content, which is the residual introns that participated in the transcription process. Here another illustration, you have the exon 1 intron, exon 2 intron, exon 3. The introns have been removed after RNA processing, and what remains has been spliced. Of course, don't forget about the 5' cap, which is necessary for protection against decay, plus adding like a header, a title, so to speak, so that this block of RNA can be recognized and also protected. Also, the poly A tail, which is the footer or the signature. R' untranslated region is here, and 5' untranslated region because it doesn't need any translation. It needs capping and tailing, respectively. Translation, the ribosomes. Okay, ribosomes are molecular machines that produce or synthesize proteins based on the mRNA sequences. Okay, by reading mRNA sequences, which are like the recipe from which we are fabricating or cooking the protein. The ribosome composition, you have large submit, small submit. Translation rate is given by U translation equal to number being translated in respect of time. Number of amino acids or like bases being translated in respect of time. tRNA or transfer RNA molecules and codons. tRNA molecules carry specific amino acids and recognize codons on mRNA. Codons, by the way, are blocks of code that are constituted of a sequence of bases, okay? Sequence of amino acids. So, they recognize codons on mRNA through their anti-codon region. The codon length is equal to 3 nucleotides. So, they are processed by blocks of 3s. Each codon is formed of 3 sequential amino acids, 3 sequential bases. The number of possible codons is actually 4 to the power of 3, okay? Which is equal to 64. Anti-codon pairing is given by codon mRNA coded into anti-codon tRNA. To give you an insight into the math of the number of possible codons, since we brought it up. Okay, why 4? 4 because you have A, T, C, G. 4 bases. They need to be grouped into 3s, correct? 3 locations, which is each codon has a placement for 3 bases. And how many, the question is, how many combinations with repetition can we get? It's not a permutation, it is combination with repetition. How many possibilities can we get from that? So let us count them. Okay, we'll try to count them. So you have A, T, C, A, C, G, O, ATC and ACT also. ACG, AGC. Then you have, we start with T, TAC, TCA, TCG, TCG, and TGC. Also, you have TAG, TGA, correct? And TAC and TCA. Okay, the point is, if you make all the combinations possible, you get 64. And this is how we mathematically get to it. So, it is n to the power of p, I think. P is the number of placements. So, you have how many placements there is, how many locations. You have 3 placements, 3 bases. Okay? So this is combination with repetition again. So you have 3 placements, and how many possible bases are there? You have the total of 4 bases. 4 to the power of 3 gives us 64. Just by the way, like when you have bits 0 and 1, okay, in the binary, so how many possible combinations can we get from 3 bits, 3 placements? It is the number of bits, or all the possible bits are 2 to the power of 3. You have 3 placements, okay? 2 to the power of 3, 8. Okay, so what are these placements? You have 0, 0, 0, 0, 0, 1, 0, 1, 0, 0, 1, 1, 1, 0, 0, 1, 0, 1, 1, 1, 0, and 1, 1, 1. These are all the possible combinations. And you have 5, 3, it is 8. By the way, if you're wondering how I got that, how I flipped between them, you keep adding 1, but here 1 plus 1 gives us 0, and you have a return. This is XOR operation, okay? Don't need to go into it, but yeah. So you have 1 plus 0, 1. Anything plus 0 gives us, yeah. You have 1, only 1 plus 0 and 0 plus 1. It's not a plus, it's XOR operation. 1 XOR 0 and 0 XOR 1 gives us 1, but 1 XOR 1 gives us 0 with a return, just like in addition, in classical addition, and 0 XOR 0 is 0. So here, for example, 1, 0, 1. You add 1 here to the right, so it gives us 0, and you have a return to the left, so it gives us 1, 1, 0. Because 1, 0, 1 is 0, and you have a return 1 here, which is added to the 0, so you get 1, 1, 0. And you keep going like this. Genetic code. The genetic code defines the mapping between codons and amino acids. Properties: degenerate, so you have multiple codons encode the same amino acid. Universal, with minor exceptions, and non-overlapping and comma-free. Okay, they don't overlap. Start codon, AUG, which is the methionine. Okay, so adenine, uracil, guanine. Remember, okay. Stop codons, it's the UAA, so this is where the operation stops. UAA, uracil, adenine, guanine, and UGA, uracil, guanine, adenine. Protein synthesis steps. Initiation, the ribosome assembles at the start codon, AUG, and the initiator tRNA binds. Okay, so ribosomes assembles at the start codon, AUG, initiator tRNA binds. Initiation probability, P init, is directly proportional to ribosome binding efficiency. Elongation, amino acids are sequentially added to the growing polypeptide chain, correct? Peptide bond formation is given by AAN plus AAN plus 1, gives us AAN minus AAN plus 1 plus H2O, which is water. Polypeptide length is given, or is proportional to the number of codons, actually. Okay? So number of codons multiplied by the length of a codon. Here, in terms of length, it is the number of codons. Okay? Termination, translation stops when a stop codon is reached and the completed protein is released. Termination efficiency is eta term is equal to number of completed proteins divided by the number of initiated translations. Energy requirements, both transcription and translation require energy. Okay, so you have ATP, adenosine 3-phosphate, and GTP, guanine 3-phosphate consumption, which are the energy fuel, so to speak. The total energy would be proportional to the number of bonds being formed. Approximate energy cost per peptide bond is proportional or approximately 4 high energy phosphate bonds. Gene expression efficiency, the overall efficiency of gene expression depends on transcription and translation rates. Protein output is proportional to the rate of transcription, proportional times actually the rate of translation. Finally, transcription and translation are essential processes that convert genetic information into functional proteins. Through highly regulated enzymatic mechanisms and precise molecular interactions, cells ensure accurate gene expression, enabling proper structure and function of living organisms. In this illustration, we see kind of a wrap-up or a brief explanation, not explanation, a brief or an overall representation of the process of transcription and translation that we've spoke about moments ago, which is the transcription from DNA to RNA. So how does it work? First, the RNA polymerase is the one responsible for the synthesis of the messenger RNA. It's like the catalyst behind the creation of mRNA. Here you notice the promoter region, and at the end, at the termination, you have these two helixes that were separated by a helicase being joined again. Correct? They are being joined again. So now you notice here the mRNA, you have the capping, which is the 5' cap, and the poly A tail, which carry the header and the signature, so to speak, of the mRNA. You have the exons with the introns. The introns need to be removed, okay, so that we can splice and form the protein out of this mRNA. Here in the lower image, you see the ribosome, which is the manufacturer or the plant that creates the protein out of the information stored within the mRNA. Or rather, by the way, this information within the mRNA should be translated, and in the transfer tRNA, this is the info used in order to manufacture the protein. You have the amino acids, the peptide bonds, and from the peptide bonds, you get the finished protein. Of course, don't forget about the starter codon and the stop codon, which would be the marker for the ending of the protein manufacturing process. On a final note, when or while the helicase enzyme is the one responsible to unwind the double strands of the DNA for the manufacturing of the mRNAs through the polymerase, so it is the one that splits both strands of the DNA molecule, you have the ligase, another enzyme, which is the one that wraps it up again to reshape, okay, to restore the bonding between both strands of the DNA so that it can recover its original spiral form, so to speak, which is the storable form within the chromosomes. Chapter 6, Mendelian Genetics. Mendelian genetics describes the fundamental principles of inheritance based on the pioneering experiments of Gregor Mendel, which is by studying pea plants, Pisum stavium, Mendel established laws that explain how traits are transmitted from parents to offspring through discrete units called genes. Law of segregation. Law of segregation states that each individual carries two alleles for a given gene. And these alleles separate during gamete formation. Okay, when you have a crossing between a male and a female. So these alleles would separate during the gamete formation to form the baby, such that each gamete carries only one allele from each parent. If an organism has a genotype capital A, lowercase a, for example, then the possible gametes would be PA, probability for A would be 50%, and probability for lowercase a would be also 50%. So a child would get either of the two alleles. During fertilization, you would get the possible combinations would be AA times AA, so they become capital A, or capital A, lowercase a, capital A, lowercase a, or lowercase a. The genotypic ratio 1:1, phenotypic ratio 3:1, and I will explain it in a moment. So this is what we're talking about. You have A, capital A, lowercase a, for the male, let's say, and for the female, you have also capital case A, lowercase a. What are the different combinations? You have A, capital A, capital A. So we are multiplying. Capital A, lowercase a, capital A, lowercase a, and lowercase a, lowercase a. So what are the genotypic ratio? 1 to 1. So you have 1 for the AA, okay, 1 occurrence, 2 for the A, lowercase a, this is the 2, and 1 for both lowercase a, correct? These are the genotypes. What is the difference? So these are the different combinations that we get. Meanwhile, the phenotypes, meaning for the physical traits that would appear in the child in the next generation, it depends on the dominant trait. You notice here we have lowercase and uppercase. For the uppercase A, it represents a dominant trait. Let's say the color of the eyes, okay, being brown is dominant. And lowercase a is the color of the eye is blue, being recessive. Since the brown eyes, so again, A is equal to brown eyes, it is dominant, and lowercase a is recessive, which is for the blue eyes. Okay, so why is it 3 to 1 here? Because every time you have a dominant gene, a dominant allele, this is the physical trait that would show up. So in those 3 first cases here, okay, 1, 2, and 3, since you have at least one capital A, the children would have brown eyes. Why? Because A, capital A, is the dominant trait. Only in one case, which is, by the way, in such case, 25%, okay, which is lowercase a, only in this case, when a child happens to have lowercase a from both of the parents, since it is recessive, only then that the child would have blue eyes. That's why for the phenotype ratio, it is 3 brown to 1 blue. Only for one possibility out of 4, you would have blue eyes in the child. Meanwhile, for the other traits, for the other 3 possibilities, you would have brown eyes, since capital A is the dominant trait for brown. So whenever you have a multiplication of the dominant with the recessive, the dominant trait would show up at the expense of the recessive one. This, of course, applies for fur, color, for not only for humans, for any type of animal, you can get the same thing, okay? Law of independent assortment. The law of independent assortment states that alleles of different genes assort independently during gamete formation, provided the genes are located on different chromosomes or far apart on the same chromosome. For a dihydroid cross, so you have AABB times AABB, gamete combinations would be capital A, capital B, capital A, lowercase b, lowercase a, capital B, and lowercase a, lowercase b, with probability for each one of them is 25%, which is 1 over 4. The joint probability is probability, let's say, of A and B is equal to probability of having A times probability of having B. To explain it in a more simplistic way, so let's say you have, for example, a bucket. You have yellow ball, green ball, and red ball. All right? So you have 1 by 1 by 1. So the probability when you take like 1 or 2 balls, okay? Probability of having, or like you are drawing one ball after the other, probability of or you're taking two balls, okay? I'm kind of, confused a little bit. So probability of drawing the red color and the green color, okay, which is PR enter G, is the probability of drawing the red color multiplied by the probability of drawing the green ball, the green colored ball. So since the probability is a quantity less than 1, probability of drawing two colors, this one and this one, okay, after you multiply it, the probability becomes less. So it becomes less, probable for you to draw these two colors. You made it hard on yourself, okay? So this is the concept behind it. Dominant versus recessive traits. Traits are determined by the interaction of alleles. So for the dominant allele, it can be expressed in both homozygous and heterozygous conditions. What we've said previously, meaning that it's enough to have only one capital A, okay, that this allele would be expressed because it is dominant. Let's say it is the color of the hair of the eyes. It would be expressed in both homozygous and heterozygous conditions. So even when it comes along a recessive allele, the dominant one, since it is dominant, it would be showing in the physical traits of the next descendant. Now, for the recessive allele, you would have lowercase a expressed only in homozygous condition. Okay, lowercase a. So only when you have the recessive gene or allele occurring twice on two alleles, this is only where the physical trait would show up. Okay, for example, for the blue eyes, only when it is you have two recessives, because with the dominant one, it would disappear. It would remain hidden. Phenotypic expression function, the phenotype is when you have a dominant gene, A would be dominant, uppercase A, both in homozygous and heterozygous alleles. As for the recessive phenotype, it would show up, meaning only if you have two lowercase a, only if you have two recessive alleles, this is when the physical trait would show up in the next generation. Monohybrid cross, so example, AA times AA, we are crossing between two parents. They have capital A and lowercase a. Okay, so what would be the result of the crossing? We've seen that before. So you have capital A, lowercase a, also capital lowercase, and two lowercase a's. The genotypic ratio is 1 capital A, 2 heterozygous A, capital A, lowercase a, and 1, 2 lowercase a's. That is for the genotypic ratio. However, the phenotypic ratio is 3 dominant, 1 recessive, meaning that the capital A trait would show up in 3 possibilities versus 1, where it's only the recessive one that would show up in the physical traits. Again, if you can imagine it being brown eyes versus blue eyes for the lowercase a, so you have here all these 3 would be brown eyes in the children, and 25% only possibility for the blue eye trait to show up. Dihybrid cross. Example, you have capital A, lowercase a, capital B, lowercase b, times or being crossed with AA, BB. Okay, so here we're talking about 2 different traits that are not, by the way, dependent of each other. 2 different traits. Let's say capital A and lowercase a are the eyes, eye color. Okay, so A, capital A, lowercase a, both for the eye color. capital A being the brown color, lowercase a for the blue color. Now for B, capital B, lowercase b, that concerns the hair color. So here you would have, let's say, blonde hair, and here you would get black hair. Okay? Actually, the blonde, usually the black is the one that is dominant. So let me change the color. Here the black, and here the blonde. So what would be the different, so you'd get 16 different possibilities here in this case. What would be the distribution? So whenever you have capital A, okay, all of the capital A is a dominant type. So 16 minus 4, basically 12, dominant. Okay? Also for the capital B, the same thing. So how many capital B that you have? B, also 12, okay? Then you'd have the lowercase capital something, okay? So you've got the point, okay? For the A, how many? Like the lowercase a and lowercase b, you have 1, okay? For lowercase a alone, you would get another 1. So lowercase b, another 1. Okay, yeah. For lowercase b, you have 2. For lowercase a, also you have 2. These, or you have 3 actually. So I hope it's right. Yeah, it's probably wrong, but you get the point. Key ratios. For the monohybrid ratio, you have phenotypic ratio 3:1. This is the first example we've seen, where you have capital A, lowercase a. The genotypic ratio 1:1. So for the phenotypic ratio, you have 3 dominant, 1 recessive, correct? Wherever you have capital A, you have dominant, dominant, dominant. Capital A, capital A, capital A, lowercase a. Always the capital A is dominant, so it is 3:1. The genotypic ratio is all the combinations. So you have capital A, lowercase a, which is 1 by 1, and 2 capital A, lowercase a. So that's the difference. Now for the dihybrid ratio, phenotypic ratio is the following. So you have 9 for the expression of A and B being dominant, I think. So 1, 2, 3, 4, 5, 6, 7, 8, and 9. You see? 9 for A and B both being expressed dominantly. Okay. So you have 4, 5, 6, 7, also 8, and 9. This is the first line. 3, I think 3 for A being dominant, B being recessive, which is 1, 2, and 3. Another 3 for A being recessive, B being dominant, which is 1, 2, and 3. Finally, 1 with both A and B recessive, which is this one. This is how we're counting it. Probability approach. Inheritance can be analyzed using probability theory. You have the multiplication rule, which is probability of having A and B at the same time. being represented in a descendants is probability of A multiplied by the probability of B. Addition rule, probability of A or B, so one or the other, is equal to probability of A plus probability of B minus probability of A and B at the same time. We're going to see an example, by the way. So here you have probability of A is 1 over 4, probability of B 1 over 4, let's say. Probability of A and B is 1 over 4 multiplied by 1 over 4 is equal to 1/16. And we're going to see also, going back to the other, the hybrid phenotypes, we're going to see the probabilities in that other example. Let's say we want to evaluate the probability of having A and B dominant, okay? That would be probability of A and B equal to probability of having A dominant multiplied by probability of having B dominant. So it is equal to probability of having A dominant. It is all of the parts that have A, correct? So this is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. This is actually all except these 4 here, okay? So it is 12 divided by 16 multiplied by the probability of having B dominant, which is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Also 12 divided by 16. So it is 3 divided by 4 times 3 divided by 4. This is a reduced form. Okay, so it is 9 divided by 16. All right? 9 divided by 16, so more than 50%. Okay, so, yeah, it's around 55%, something like that. Now, probability of having A being expressed in the phenotype or B. So one of them is okay. That is probability of A being expressed plus probability of B minus probability of A, lowercase a, enter B, meaning having A and B at the same time. So it is equal to probability of having lowercase a. This is only when you have 2 A's, okay? 2 double A's, which is 1, 2, and 3, and 4. Anything else? No, okay. So it is 4 over 16 plus probability of B, lowercase b, being expressed. This is only when you have this one, 2 B's, 2 lowercase b's, this one, okay, and this one. Also 4 divided by 16 minus probability of A enter B, which is minus probability of A enter B. Probability of A and B at the same time. What is the probability of having A and B at the same time? It is this one, which is 1 over 16. Only when you have double lowercase a, double lowercase b. So minus 1 over 16. So it is 8 over 16 minus 1 over 16, which is equal to 1/2 minus 1 over 16. Correct? So 0.5 minus something, yeah, approximately, let's say 0.48, I don't know, 47%, let's say. Okay, this is how you evaluate it. Test cross. A test cross is used to determine the genotype of an individual with a dominant phenotype. So A being test crossed with double lowercase a, if offspring ratio is 1 to 1, then A to A. Okay, so meaning that this is 100% dominant, and it is the uppercase A is the dominant one. Limitations of Mendelian genetics. Mendelian laws apply under specific conditions where genes are unlinked, so they are not interdependent. Genes should be independent. They cannot affect each other, because you might have a case where 2 or 3 genes can be codependent, affecting the same trait. You need a complete dominance, which means that one allele should be dominant, the other is recessive. You cannot have 2 alleles that are co-dominant, or partially one is dominant against the other, meaning that some of the traits of the recessive can still appear in the descendants. And no epistasis or gene interaction. So the genes should not be interacting with each other to affect the final outcome. Deviations to Mendelian genetics occur in incomplete dominance. What we mentioned, that when you have one dominant and one that is partially recessive, meaning that some of it or some of its trait can still appear next to the dominant one. So there's still a mixture. The recessive one is not completely disappearing. It's not completely but partially recessive. Second, codominance, meaning that you have both of the alleles are dominant, such as, for example, you have red flower and white flower. Both of the traits are dominant, so the crossing between both of them would produce a pink flower, which is the mixture of both of the traits. And linked genes, meaning that the genes are linked together. They can affect the same trait. They are dependent. So this can change the Mendelian genetics into a completely different ball game, so to speak. Here in this example, so you have parental crossing, purple flowers, white flowers. The first generation would get, okay, all purple flowers. Why is that? Due to the fact that the purple is dominant. Okay, that would be the law of segregation, affecting and being affected by the dominance. Another generation, if you have two having received the recessive trait, which is lowercase p, the white flower. So white flowers would show up if you have 2 recessive alleles that are white. In case of a purple one, wherever you would have a dominant allele, you would get purple flowers. So here you see the combinations, p lowercase p, you would get pp, p lowercase p, and lowercase p. So only 25% probability or possibility of having white flowers. The other 75% is for the purple flowers due to the dominance of the purple trait. Another example for that, you have pure green peas and pure yellow. So capital Y and lowercase y, okay, being crossed together. First generation would give you all green. Now, the second generation, you would start getting yellow, okay, which is supposedly, you'd start getting green, which is supposedly the pure green recessive trait, but it will start showing up because it still have a probability of occurring. Here you see 25% probability, okay? So this one, when it crosses or intermarries with something lowercase or uppercase y, or if it's completely recessive, it can still produce recessive trait, which is the green. Finally, Mendelian genetics provides the foundational framework for understanding inheritance. Through simple probabilistic rules and allele interactions, it explains how traits are transmitted across generations and serves as the basis for more advanced genetic studies. Chapter 7, Non-Mendelian Inheritance. Non-Mendelian inheritance encompasses patterns of genetic transmission that do not follow the classical laws established by Mendel. These patterns arise due to more complex interactions between alleles genes and environmental factors. They play a crucial role in explaining the diversity of phenotypes observed in nature. Starting with incomplete dominance. Incomplete dominance occurs when the heterozygous phenotype is intermediate between the two homozygous phenotypes. Example, RR red crossing with RR white would give us pink. So it's not a complete dominance. Capital R, lowercase r, which is a mixture between the red and the white, would give us pink, instead of the red classically being dominant in such case, giving us red color. Phenotypic ratio is 1 to 1. Why? Because you will get 2 RR, which are capital case R, lowercase r, the pink colors, one being completely purely red, which is capital R, and one lowercase r being white. Let phenotype value be represented quantitatively. PRR is equal to, which is probability, okay? It is P capital case R plus, capital case R plus P lowercase r divided by 2. Codominance. In codominance, both alleles in a heterozygote are fully and simultaneously expressed. Example, ABO blood group. So IA, IB would give us type AB. They are both represented, okay? So both of them remain. Phenotypic expression is given by P is equal to probability of A plus probability of B. No blending occurs. Both traits are visible. Multiple alleles. A gene may have more than 2 allelic forms within a population, although an individual carries only 2. Example, ABO blood group systems. Okay, so IA, IB, lowercase I. Genotype combinations, you have IA, lowercase I, IB, etc. Allele frequency constraint is given by P plus Q plus R is equal to 1, with P is the frequency of IA, Q that of IB, and R is the frequency of I, lowercase I. Polygenic inheritance. Polygenic inheritance involves multiple genes contributing additively to a single trait. So this results in continuous variation. Phenotype model is the sum of different genotypes of different genes plus E, which is the environmental effect. So P is equal to sigma I equal 1 to N of all genes indexed by I plus E, which is again the environment effect. For two loci, A and B, number of contributing alleles would be capital N is equal to lowercase n-A plus n-B. Phenotypic distribution approximates a normal distribution. Px is proportional to n mu sigma square. Number of phenotypic classes is given by 2n plus 1. Lowercase n is the number of gene pairs. Epistasis. Epistasis occurs when one gene masks or modifies the expression of another gene. Example in recessive epistasis. Modified phenotypic ratio is 9:3:4. General interaction is P is equal to the phenotype is a function of the genotypes, of course, where the expression of G2 depends on G1. So G1 and G2 are interdependent. They affect both of them, okay, the physical trait in the phenotype. Probability modification, probability of phenotype is equal to sigma, probability of genotypes contributing to the phenotype. Pleiotropy occurs when a single gene affects multiple phenotypic traits. So if one gene, G, influences multiple traits, you would get G is influencing T1, T2, T3. Different traits are dependent on that very same gene. So it's one gene but affecting multiple traits at the same time. Phenotypic vector, P is equal to P1, P2, all the way to Pn. Example relation is Pi equal Fig, meaning that the phenotype is a function of the genomes that affect that phenotype. Or in other words, to be more accurate, this gene is affecting, through a specific functionality, multiple phenotypes, multiple traits at the same time. Comparison with Mendelian inheritance. Unlike simple Mendelian inheritance, phenotypes may not follow discrete ratios. Gene interactions can alter expected outcomes, so the math behind it becomes more complex, so to speak, and environmental influence is often significant. It plays an important role and it is an important factor in the outcome. Deviation measure, delta is equal to P observed minus P expected, which is the Mendelian probability. Finally, in this wrap-up kind of illustration or representation, graphical representation, we see the different characteristics of the non-Mendelian genetics and how they go against violating the laws, the classical rules instated by Mendel within the Mendelian genetics. Okay, so what are these? First, you have incomplete dominance, where red and white, for example, here, which are two traits for the flowers, when crossed among each other, they can produce a pink color, which is where both the red and the white can stand in the next generation. So there is no dominance. Both the red and white are dominant and they mix up to become a pink color. So both of them would be expressed. Second, codominance. In codominance, you would have, instead of a mixture, both of the traits would show up without any mixture, meaning that you have a brown hamster and a white hamster. Okay, so the descendants would get both the brown and the white. It wouldn't be like a mixture, a lighter brown. No, they would get both traits within their fur. So you have the white here, the brown, and both the brown and the white being expressed in one of the children. Both traits are being expressed. Multiple alleles, so you have A, B, B, so they still exist and they are still like they carry on in the next generation, so they are not taken over by another. All of them would be expressed. They are kept up in the next generation. They show up. And finally, polygenic inheritance. So you have here different mixtures of colors when it comes to the skin color, for example. So you get different descendants. As a conclusion, non-Mendelian inheritance expands the understanding of genetic complexity by incorporating gene interactions, multiple alleles, and environmental effects. These mechanisms explain the wide range of phenotypic diversity observed in biological systems, which cannot be explained solely by the Mendelian genetics. And these are essential for modern genetics, medicine, and evolutionary biology. Chapter 8, Mutations and Genetic Variation. Mutations are permanent changes in the genetic material, DNA sequence, of an organism. They are the primary source of genetic variation, which is essential for the evolution, so to speak, adaptation, and biodiversity. While some mutations are harmful, others can be benign or neutral or even beneficial. Types of mutations. So, you have point mutations. Point mutations involve a change in a single nucleotide base, general form. So A, for example, flipping into a mutated G from adenine to guanine. Types, substitution, one base is replaced by another. Silent mutation, no change in the amino acid. Missense mutation, different amino acid is produced. And nonsense mutation, so premature stop codon is introduced. It is introduced prematurely before the manufacturing of the protein. The mutation rate per base is given by mu is equal to number of mutations divided by the number of bases. This gives you the mutation rate. So in other words, you have 10, 2 have mutated, that would be 2 divided by 10, which is 1 divided by 5, or 20%. Insertions and deletions. or indels, insertions and deletions involve the addition or removal of nucleotides. You have sequence shift, it is called also frame shift, if delta n is different than 0 modulo 3. Effect on protein length, so n prime AA is equal to n plus delta n, which is the number or the change in the nucleotide divided by 3. Okay, so how many nucleotides have changed, that would affect the protein length. Frame shift mutations alter the reading frame, potentially changing all downstream amino acids. So it can spill over in the sequence. It can affect the entire sequence, which can be very dangerous. Chromosomal mutations. Chromosomal mutations involve large-scale structural changes in chromosomes. You have duplication. A segment of chromosome is duplicated, so segment ABC becomes ABC. Gene dosage effect, expression is proportional to the copy number. Inversion, meaning that a chromosome segment becomes reversed, so ABCDEF becomes ABEDCF. A flipping. Translocation, which means that a segment from one chromosome is transferred to another segment, such as reciprocal translocation, so A1B1 plus A2B2 becomes A1B2 plus A2B1. The causes of mutations. What are the different causes? I can cite poisoning or radiation here. Let's start with radiation. High energy radiation such as UV, X-rays can damage DNA because it is called desoxyribonucleic acid. So any type of radiation can break these bondings of hydrogen bonds between different bases. So if they break, they would reconnect to other kinds of bases. That would cause a mutation. Okay, this is caused mainly by the breaking of chemical bonds again. Okay, the damage probability is proportional to the radiation dose to which the DNA or the organism is being exposed. Chemical mutagens. Certain chemicals alter DNA structure or base pairing. Mutation induction rate is given by mu induced is equal to mu0 plus delta mu chemical. Examples, you have base analogs, alkylating agents, and intercalating agents. DNA repair mechanisms. Cells possess sophisticated systems to detect and repair DNA damage, maintaining genomic integrity. You have the mismatch repair. It corrects error introduced during DNA replication. Error correction efficiency is given by eta equal to the number corrected divided by the overall number of errors. Base excision repair or BER. This, by the way, looks like the BER, bit error rate acronym within when it comes to data transmission or communications, electrical communications. Base excision repair or BER removes damaged or incorrect bases completely. The repair rate is equal to the number being repaired in respect of time. Nucleotide excision repair or NER removes bulky DNA lesions such as thymine dimers. You have double-strand break repair, includes homologous recombination, it is accurate, and non-homologous and rejoining, which is error-prone. The repair fidelity is given by the number of correct repairs divided by the number of total repairs. Again, the difference between these two repair mechanisms is that for the homologous recombination, actually, this type of repair is more accurate. Why? Because you see the cell would use a DNA sequence that is identical to the part that has broken, and this template, okay, it would be used as a template in order to repair the break. So it is more accurate, mainly used during S and G2 phase of the cell cycle. Okay, so the DNA double-strand break occurs, then this the cell would find a homologous DNA sequence used as a template in order to repair the break. As for the non-homologous and joining, so this is what would happen. In this way of fixing the problem, it would be less accurate. A quick repair mechanism is done by directly joining the broken DNA strands. So it is less accurate, okay, probably faster, okay, but the negation is like immediate. So it's broken, it's then fixed, but there is a larger probability for errors. Genetic variation. Mutations contribute to genetic diversity within populations. Total variation is given by V is equal to sigma mu I. Allele frequency change, so delta P is equal to mu times 1 minus P minus nu P, where mu is the forward mutation rate and nu is the reverse mutation rate. Impact of mutations. Mutations can have different effects. You have the neutral, so no effect on phenotype. Deleterious, meaning that a deletion would occur. It has a harmful effect because you have information blocks being lost, being deleted. Beneficial, which means that it provides adaptive advantage, such as fitness effect, W is equal to 1 plus S, where S would be the selection coefficient, so to speak. So what is a mutation? A mutation is a permanent change in the DNA sequence. Mutations are the ultimate source of genetic variation also. So they have a beneficial aspect. They are not always harmful. Chromosomal mutations, so you would get inversion, meaning one part of a strand goes to the other strand, of chromosome here. Duplication, so a segment would be repeated. That can be very dangerous. Types of genetic mutations, substitution, so you have here A being flipped into T, for example. Insertion, A being or a G being inserted between A and T, correct? And deletion, so one amino acid would be lost, one base would be lost. Insertions and deletions or indels, so this is the original. Insertion, meaning B has been inserted between G and CC, or deletion, so you have one amino acid or base, so to speak, have disappeared, have been deleted. So this can sequentially change the entire DNA block. It can be very dangerous. Another type of mutation is the translocation. So one segment is moved to a non-homologous chromosome, and deletion at a large scale. An entire segment can be lost. Now, causes of mutations: radiation, mainly radiation and X-rays and gamma rays, or chemicals, chemical poisoning. DNA repair mechanisms, so you have proofreading, DNA polymerase checks and corrects errors during replication. Mismatch repair fixes base pair matches that were missed during replication. Nucleotide excision repair removes and replaces damaged bases, that's very important. And double-strand break repair, which we've seen moments ago. The homologous and non-homologous type of repairs. Why mutations matter: single cell mutation, genetic variation, diversity in population. So this can be a beneficial trait in organisms. Here another illustration if you want to check it. So genetic variation. Here you have causes such as radiation, chemicals, viruses. Types of mutations: point, mutation, insertion, deletion, etc. Effects on mutation: it can be harmful, neutral, no effect, or beneficial. So there might be, I believe, there's a mechanism for mutation which is built in the genome, in the architecture of the cell, in order to produce like new varieties of a species. At the end, mutations are the fundamental source of genetic variation, driving evolution and diversity, while they can disrupt normal biological function. Cellular repair mechanisms work to minimize their harmful effects, maintaining the stability of the genome. Finally, thank you for watching and see you next time. I hope that you enjoyed this video.